Hey everyone, my name is Joseph and I'll be taking you through the introduction to the disturbances of cell metabolism and concretions. Yes, so let's first understand when someone talks about cell metabolism, what do they mean? So cell metabolism are basically all the chemical changes that take place in their cells. And some of the other terms that we're going to see are parenchymal cells. So parenchymal cells they are basically primary cells in the tissues that basically perform the primary function of these tissues, and they are found in tissues such as the liver, heart, lung, and the brain. When we are going through this, at the end, we would wish to be able to understand or appreciate some of these objectives as put forward. The first one is to enable you to recognize and identify the abnormal depositions intracellularly and extracellularly as a result of abnormal cell metabolism. This is basically the introduction starting off, but then also more sessions are to come, so stay on the watch as the new videos are actually coming up from my other colleagues. We shall also appreciate the description of how best can one actually describe the microscopic and the macroscopic features of the abnormal deposits, or you would call them concretions, and also to understand the mechanisms through which these concretions actually develop. Now, let's kickstart with the first one, which is going to be about the abnormal intracellular accumulations in the cells. I'll cover the first one, which is fatty change. Call it, or it's the lipidosis. So, fatty change, when someone hears the word fatty change, you're looking at, there is probably accumulation of fat in one or the other. That should be actually your first thinking, accumulation of fat in the cells. Now let's appreciate it in a proper scientific definition. Fatty change refers to the abnormal accumulation of fat in the cytoplasm. It's accumulating in the cytoplasm of the parenchymal cells, especially the liver, heart, kidney, and the muscles. We appreciated this earlier. So that is fatty change. You're having abnormal accumulation of fat in the cytoplasm of parenchymal cells. This accumulation of fat would basically will occur due to the imbalance in the production. If there is an imbalance of production, a lot of production is occurring, that would probably result into fatty change. Either or you're looking at utilization is not sufficient, there is a lot but it's not being taken up, or we are looking at mobilization of the lipid precursors within the affected cells. So that is how best it actually comes about. And fatty change is reversible and indicates significant cell injury. It may follow acute cell swelling or occur separately. So this is something, if there is acute cell swelling, it would occur, or it can also occur independently or separately. Now, what is the pathogenesis of fatty change? How does fatty change occur? How does it develop? Fatty change can be best understood when we're talking about the liver, because in the liver we have the triglycerides that are incorporated. The triglycerides are incorporated to proteins as lipoproteins, and then they are released into the cell. They are released from the cell to the plasma. So that is now going to be flowing in blood. So fatty liver results from the imbalance of any or several of the metabolic steps that are involved in these lipoprotein formations. Now, what are these? Now, this is the real guest. This is the real pathogenesis of how fatty change occurs. The first, in the liver, there are basically 5, and we should be able to actually appreciate them and remember them at any one point in time. When we're talking of fatty change, the pathogenesis, these should actually come. The first one is the excessive delivery of fatty acids. to the liver. That is the first one. The second one is if there is a blockage of fatty acid oxidation to ketones. The third one, if there is impaired synthesis of apoproteins. The fourth is if there is impaired combination of the lipids and the protein. And the fifth is the impaired release of these lipoproteins. Now, let's just dig deep down into. If there is excessive delivery of free fatty acids to the liver, these could result into increased triglycerides synthesis. Lipolysis occurs, so lipid breakdown would occurs in, first of all, starvation, because you're trying to mobilize more energy so that someone cannot actually die out. So you're going to look at breaking now the secondary storage sources. And the secondary storage sources, they are lines which are first broken down, and the lipids is the first one because it has the higher energy. Then in lipolysis could occur if there is inanition, and inanition is the inability to swallow food. Someone with diabetes mellitus can also have lipolysis, and in ketosis and hyperadrenocorticism. So that is how we are going to actually have excess delivery of free fatty acids to the liver, because we are actually looking at mobilizing of energy in one way or the other. Now, if there is blockage of fatty acid oxidation to ketones and other bodies, that might result from mitochondria injury, especially that is induced by the toxins, or hypoxia. If there is low oxygen, that would result into mitochondrial injury. If there is impaired blood flow and hepatocellular hypoxia, that would also still bring about the blockage of the fatty acid oxidation to ketones. Impaired synthesis of apoprotein. So you're looking at the lipid acceptor proteins. So remember, if there is because the protein is supposed to come and combine with the lipid. So if there is an impaired synthesis of this apoprotein, which is responsible for accepting the lipids, then that would also result into free fatty acid accumulation, would result into fatty change. This normally occurs if in chloroform and also in cases where we have aflatoxcosis. Impaired combination of the lipids and the protein. This is something that is not very common as well. The impaired release of the lipoprotein is also not so very common, but these are cases that can actually result into fatty change. Remember, the first one is if there is excessive delivery of free fatty acids to the liver, that would result from triglyceride synthesis, and that normally occurs when we have lipolysis occurring, which would now cases or scenarios where we have lipolysis occur is in starvation, inanition, diabetes mellitus, ketosis, and also hyperadrenocorticism. If we have blockage of fatty acid oxidation to ketones, where we are also seeing the injury of the mitochondria, also impaired blood flow and hepatocellular hypoxia, those are cases that would result into fatty change. If we have impaired synthesis of apoprotein, which is responsible for accepting the lipid, that would also result into fatty change. Impaired combination of the lipid and the protein, and lastly is the impaired release of the lipoprotein. Now, let's appreciate how to describe or how the fatty change would look like glossly. So this is the gloss appearance of fatty liver. Now, we said one of our objectives is one needs to actually appreciate the microscopic and the macroscopic view of actually these cell metabolism disturbances, but also we need to be able to describe them. In pathology, if you cannot describe, then if you cannot tell me what you see, then there is a problem. You need to be able to tell me what you see, then you can actually proceed. That is the major art in pathology. So when you look at this liver here, this liver is basically, first of all, what we see glossly is that this liver is enlarged. Now imagine you have something that is actually filled with fat. Already it is enlarged because there's going to be now we have intracellular additional depositions, so it actually causes its size to increase. Where we have this liver is actually basically soft, and when you cut through, it is greasy. The color of this liver is yellow tan color. It's a yellow tan color, if you appreciate that. Now, moving further, the centrolobular hepatocytes are severely affected. That is, if we go now to the center of the liver, the centrolobular hepatocytes are more severely affected. That is something that you appreciate now, that we shall appreciate more in the microscopic view. When I'm describing this liver, I'm going to begin with the color, how does it look, and then I begin describing the lesion, how is it, and where are they found, and then it is also very key to identify which specimen this liver was actually taken from. The specimen from which the liver was taken from is basically dog. That is due to the multilobular format or anatomy that we are appreciating. And when we begin describing, we begin with the color. This is a pale brown or yellowish liver, which is diffusely swollen all over. It is swollen with multiple round white spaces, if you appreciate these spaces, that are distributed throughout. So that is a perfect description for this liver. When we come to now the microscopic appearance of the liver, of the fatty liver, we are going to also see what we see here is that it is affecting every cell. All cells are affected, if we see from this view, because you are looking at the white droplets that are appearing all over. It's more or less like a sky with the stars. So affected cells have numerous clear round vacuoles. These are round vacuoles in the cytoplasm. In very fatty cells, the nucleus, if you appreciate this nucleus, has been pushed to the peripheral. Yes, it has been displaced to the sides, and the cytoplasmic space is enlarged and clear. So this is all cytoplasmic space, which is enlarged and clear, and the cytoplasm have been pushed to the sides. Just something that we wouldn't want to confuse is the difference between the lipid vacuoles or the fat vacuoles and also the glycogen degeneration, which someone will actually take us through. If you look at the cells, the nucleus are still in the center, though the cells actually appear to be whitish. The background is whitish, so there is extracellular deposition of glycogen within this liver. Yes, so, and if you look here, they've been pushed to the sides. Now, let's discuss or share something about fat infiltration. So, fat infiltration is, we are having extracellular fat accumulation out of the cell, out of the cell fat accumulation. So, fat infiltration is the same as steatosis. So, and it refers to the accumulation of adipose tissue where they are not usually present. So you are seeing that fat in areas that it's not usually present. In most cases, we are seeing it in the extracellular areas. So it's extracellular abnormal fat deposition, and fat cells often fill the voids or created by the lost cells or tissues. In case we have a tissue or a cell that has died, it's easy for the fat to actually invade that area and it will deposit. So the lesion occurs in cardiac, skeletal, and connective tissues of the pancreas and the lymph nodes. So that's where we normally see this fat infiltration. When I'm to describe this, what I'm seeing pathologically, basically I'm seeing white spaces in the muscle bundle, because this, you appreciate this as skeletal muscles, in the muscle bundle, or adipose tissue in the muscle tissue. So thank you very much for tuning in and listening to us. That has been it about the introduction, and we're going to see more of the sessions coming through from my colleagues. Thank you.